Finishing writing a thesis and finishing. We have a course that will teach this term. Hi, Nat. How are you?
Where she was born and she went to school. In her high school years, she was more newspaper editor, word kind of person, and but found her way to Penn State to study chemical engineering, and Carnegie Mellon for more engineering, chemical engineering, and spent a few years with General Motors doing engineering, as you might expect, and then 15 years at Gillette in South Boston, which is sort of the sister hospital to Boston College, right here. At Watertown High School, he excelled in running away from things, track, cross country, all that. He's since been studying the extracellular matrix biology and biochemistry, and he's working on proteoglycans that modulate protein activity. And so I'd like to invite Matthew to give us his talk on computational analysis of heparan sulfate protein binding specificity.
Right? And hopefully this would be a nice dessert. Remove the dessert that you're forced not to have at the moment. So our lab's been really interested in these, or fascinated by these molecules, the heparan sulfate proteoglycans. Fascinated and occasionally frustrated, and often, I'd say, also always humbled by their amazing complexity that I'll talk about for those who are less familiar with them. Because they are in fact so complex, because they are not template driven, they present some really unique challenges to analysis, especially for traditional biochemists, where we like to reduce our problems to very simple things. But Jean has been able to tame her computers and without smoking. So many of you probably have the same relationship I have with my computer, but hopefully we'll see by the end of this lecture that there are some sort of valuable ways in which computational methods can be used to help us identify some important structure-function relationships in heparan sulfates. So this group probably doesn't need this introduction, but it's always better to be safe than sorry. Some of you probably do. So heparan sulfate proteoglycans, or as I'll often refer to them as HSPGs, and sometimes when I talk about just heparan sulfate, I call them HSs, are these really complicated, fascinating molecules. They are complex macromolecules made up of a core protein, a protein with covalently attached linear chains of heparan sulfate. One of the principal functions that we and many others in the field believe is through binding those proteins, they can assist, modulate, manipulate protein-protein interactions. Okay? So the famous one is the way in which heparin, which is a highly sulfated form of heparan sulfate, and there's 4 different enzymes that can do that. Some of these glucuronic acids get polymerized into iduronic acid through the action of this enzyme. With proteins, we know what we're dealing with. Proteins are really well behaved, manufactured from precisely defined instructions. Template, read from the DNA, we can know the primary structure of the protein, so that they're actually consistent. So unlike our McDonald's cheeseburger, we might think of heparan sulfate as being something that's sort of made in a very nice, fancy restaurant to get this nice entree, like our food tonight. So you might say, which one do you want? Well, obviously you want that, right? No, maybe not. You know, there's sort of a really nice thing about consistency. As you discover a new protein, you know what to do. Pretty classic. Find a new protein, I could write the grant probably as I drive home from work in a recorder. You want to isolate, purify, you see it's clone, you express it. Well, you can do bulk isolation of heparan sulfate. That's the kind of thing we do, and a lot of people do. We've been doing that for 20 years. And we can analyze some general structural properties, average chain length, polydispersity, maybe disaccharide, the presence of those in our 100 chains, see how evenly they're spaced, see how different they are. Any motif you want let's say you have a hypothesis, and this is a lot of hypotheses about chemical literature, that certain structures bind certain proteins. I'll just show you some experiments that we've done in cell culture toward that end, which is to ask this question: does heparan sulfate limit elastolysis? That's the ability of this enzyme elastase to chews up its protease, chews up elastase. The idea that their structure is, I think, they're very stable. Right? So, unlike a protein with a three-dimensional folded structure, I think the structure is really basic chemistry. I mean, the sulfates. Joe and Kathy's really shoot huge electron beams on them, and the sulfates will pop off. But I think so that's my dream.
You really like the way it is. So you will go. All right.